Welcome to this lecture on gas dissociation curves. Today, we are going to explore one of the most elegant and vital life-sustaining systems in the human body, the oxygen delivery system. We will understand exactly how your blood knows when to hold on to oxygen and when to let it go, ensuring your cells survive and thrive. To begin our journey, let us imagine you are running a highly efficient package delivery service. In this analogy, your delivery trucks represent a special protein in your blood called hemoglobin, and the packages they carry are oxygen molecules. When the truck visits the main warehouse, which represents your lungs, there are thousands of packages everywhere, so the truck easily fills up to maximum capacity. However, when the truck drives out to the neighborhoods, representing your body's tissues and muscles, there are very few packages around. Because the neighborhood is low on packages, the truck's doors automatically open, dropping off the oxygen exactly where it is needed most. Now that we have a conceptual picture of our delivery trucks, let us translate this into formal physiological terms. The gas dissociation curve is a graphical representation that plots two key variables against each other. On the horizontal axis, we have the partial pressure of oxygen, written as PO2. This is essentially the concentration or physical push of oxygen gas dissolved in the blood plasma. On the vertical axis, we have hemoglobin saturation, written as SO2. This tells us what percentage of the available binding sites on the hemoglobin molecules are currently holding on to oxygen. Together, these variables map out how oxygen gets picked up and dropped off. Understanding these variables leads us to a fascinating observation about the graph itself. Unlike a simple straight line, this graph has a very distinct S shape, which scientists call a sigmoidal curve. Why does it look like this? This S shape occurs because of a remarkable biological phenomenon called cooperative binding. Hemoglobin is a complex protein, and when the very first oxygen molecule binds to it, the protein actually changes its physical shape. This structural shift makes it much easier for the second, third, and fourth oxygen molecules to attach. They cooperate. This means hemoglobin becomes increasingly hungry for oxygen once it starts binding it. This brings us to an important question. Why does this specific S-shaped curve matter so much to our survival? If the relationship were just a straight line, hemoglobin would release oxygen constantly and randomly, which would be highly inefficient. Instead, the curve has a flat top. This flat top ensures that even if the oxygen levels in the air drop slightly, like when you travel to a high-altitude mountain, your blood still gets almost completely saturated with oxygen in the lungs. It acts as a biological safety buffer, guaranteeing that your delivery trucks leave the warehouse fully loaded even when supply is slightly low. While the flat top protects us in the lungs, the rest of the curve is just as crucial. Let us look at the steep middle part of the curve. This section ensures that when blood reaches active tissues that are consuming oxygen rapidly, a massive amount of oxygen is instantly released from the hemoglobin to feed those starving cells. Because the curve is steep here, even a small drop in oxygen pressure causes a huge release of oxygen. Without this precise mathematical relationship, human beings simply could not sustain high-energy activities like running, swimming, or even thinking deeply. Now that we understand the biological importance of this curve, let us break down the mathematics behind it using a version of the Hill equation. This formula perfectly models our S-shaped curve. Let us define the symbols. SO2 represents the percentage of hemoglobin that is saturated with oxygen. PO2 represents the partial pressure of oxygen in the blood, measured in millimeters of mercury. P50 is a special constant, the exact oxygen pressure required to get hemoglobin 50% saturated, typically around 27 millimeters of mercury. Finally, the exponent n is the Hill coefficient, representing the degree of cooperativity, which is approximately 2.8 in humans. Notice that if PO2 increases, the numerator grows rapidly, pushing the total saturation closer to 100%. With our formula ready, let us calculate how full our hemoglobin delivery trucks are when passing through the lungs. We are given a lung oxygen pressure of 100 millimeters of mercury. We substitute this, along with our standard constants of 27 for P50 and 2.8 for the Hill coefficient, into the equation. Following the order of operations, we first calculate the exponential terms, yielding approximately 398,107 in the numerator and 11,833 plus 398,107 in the denominator. Adding the denominator values gives us 409,940. Dividing these numbers gives a decimal of 0.971. Multiplying by 100 gives a final saturation of 97.1%. Physically, this means almost every single binding site is filled. The trucks are fully loaded and ready to travel. Now, let us see what happens when that fully loaded blood reaches your resting muscle tissues. In resting tissues, the local partial pressure of oxygen drops to about 40 millimeters of mercury. We use the exact same formula and constants, but substitute 40 for our oxygen pressure. Evaluating the exponents, 40 to the power of 2.8 is approximately 37,112. We place this in the numerator and denominator, adding it to our constant 111833 to get a new denominator of 48957. Dividing 37,112 by 48957 and multiplying by 100 gives us a final saturation of 75.8%. This means the hemoglobin saturation has dropped significantly as it passed through the tissues, releasing oxygen exactly where the cells need it. By comparing our two calculations, we can uncover the true brilliance of this system. The math tells us that the blood started at 97.1% saturation in the lungs, but dropped to 75.8% when it reached the resting tissues. By simply subtracting these values, we find that the hemoglobin dropped off about 21.3% of its total oxygen payload to feed your cells. This perfectly illustrates the dissociation curve in action. Hemoglobin holds tight in the lungs, but automatically lets go of just the right amount of oxygen when it senses the lower pressure in the tissues. To bring all of these concepts and calculations together, let us look at the actual graph of the oxygen dissociation curve. On the horizontal axis, we have the partial pressure of oxygen from 0 to 100. On the vertical axis, we have the saturation percentage. Notice the beautiful red S-shaped curve? We can clearly see the three key points we discussed. At the far right, under lung conditions, at a pressure of 100, the curve is flat and saturation is near maximum. In the middle, under resting tissue conditions, at a pressure of 40, the curve is steep and saturation has dropped to about 75%. Finally, we see the P50 point, marked at a pressure of 27, where saturation is exactly 50%. This visual map perfectly summarizes how our body adapts to deliver oxygen. As we conclude our lecture, let us review the key takeaways. The gas dissociation curve is the ultimate biological blueprint for oxygen transport in the human body. We learned that its unique S shape is driven by the cooperative binding of oxygen to hemoglobin. This specific shape is vital for our survival, ensuring maximum oxygen pickup in the lungs and highly sensitive oxygen release in the tissues. By understanding the Hill equation and the relationship between partial pressure and saturation, we can see exactly how our life-sustaining systems adapt every single second. You have now mastered how mathematics and biology work together to keep our cells nourished and alive.